Okay, great. Let's get started. So, hello everyone, and welcome back to our High-Affinity Talks series. This is where we spotlight new research in the antibody and biologic space. My name is Monica. I am your host today, and I am the head of go-to-market at My Laboratories. Many of you know us as the creators of MyXCR and Platforma, and this is our spotlight to show innovative real-world applications of our technology and others. So our speaker today is Joseph Ng. He is the lecturer and group leader at UCL. His research sits at the intersection of computational biology and immunology, where he develops machine learning approaches to understand better B cells and antibody development. In today's talk, he will introduce ImmunoMatch, which is a framework to uncover the principles behind heavy and light chain compatibility and B cell maturation. After his presentation, he will also be joined by Sam Poslawski, CEO of My Laboratories, for a live discussion. Throughout today's presentation, feel free to drop your questions in the chat, and we'll leave time at the end for Q&A. And also, this webinar will be recorded and it will be shared later this week. So, Joseph, we are so glad to have you with us today, and I will hand it over to you.
Thank you very much, Monica, and thank you very much also for the invitation in general. I think it's a fantastic idea to have sort of frontiers really of research in the sort of antibody and B cell space to be featured in a seminar series like this. So I am a group leader at UCL in the United Kingdom, and I just started my group from January. I'm working on different computational immunology problems, but specifically in antibodies, which is what I work on for quite a number of years. So let me just see if I can move the slides. Okay, fantastic. So I just wanted to start off with kind of a preface really about antibody biology. So I think regardless of which side of antibody biology you come from, whether you are more of a biologics person thinking about using antibodies as drugs, as specific binders to proteins, or you are more as an immunologist wanting to understand the dynamics of antibody response. I think we often have this idea and conception that antibodies are there because we want them to be highly specific to the antigen and high affinity, binding them to high affinity. So antibodies are more than that. Shown here is just one of these very recent work on computational antibody research, which is about sort of in silico design of antibodies based on structural data. So I think now we begin to have this idea of sort of in silico design of a highly specific and high affinity binder from scratch. We tend to only think about antibodies as, something that is highly specific and high affinity. But I think we cannot forget that there is a whole bunch of other factors concerning the function of the antibody that we need to look into. This idea about antibody design really only focus on the specificity and the affinity to it. As you can see here, this is the entire antibody molecule, and you can have a whole load of other considerations we can think of when we are trying to develop a new antibody. I think in the sort of biologic space, we tend to think of this as developability sort of considerations and measurements. So this is the sort of thing that I would like to focus more on, which is these sort of understudied aspects of antibody structure and function, really. So I'm a computational biologist. I work kind of in the middle between really developing and designing antibodies, but also to understand how the sort of natural antibody response give rise to these highly specific antibody binders. So I work a lot with my immunologist collaborators, where we look into B cell sample from different immune conditions. We together do quite a lot of molecular profiling. So you can imagine quite a bit of BCR, B cell receptor antibody repertoire work, but also increasingly looking into single cell transcriptomics as well. with the ultimate goal that we want to see whether we can understand and predict antibody properties. So this is the sort of entire pipeline that I'm interested in to sort of contribute computational methods that help in each of these processes. I will particularly focus on the molecular profiling side of it. I was mentioning we do quite a lot of single-cell transcriptomics, but I want to focus a little bit more on the sort of B-cell receptor / antibody side of the coin. So I hope I don't need to impress on you that this audience that we have both the VDJ segment that encode the variable region of the antibody and also the constant region of the antibody. So I develop computational tools that focus on both of these aspects. I have been involved in producing several tools that look into sort of sequence features of the variable region. I also look into the constant region, sort of extracting signal from data to infer dynamical processes such as class switching from single-cell sequencing data. This is not what I'm going to talk about today. I'm going to focus on the sort of last bit of the sort of entire pipeline to actually think about antibody properties. So as Monica was saying, the sort of main tool I'm going to talk about is this tool that we have recently published in Nature Methods last year called ImmunoMatch. So I hope that most of you have sort of idea about how antibodies are developed naturally in vivo. So we know that antibodies by default are highly diverse in our body. We have a huge antibody repertoire diversity, and the sort of biological mechanism that gives rise to this diversity is that you have both chains of the antibody, the heavy and light chain, and each of them has independent events of picking, essentially, DNA fragments from the genome that they randomly recombine to form this heavy and the light chain. So in the case of the heavy chain, you need one copy of the VD and J genes. So each one of them, you need to take one out of this pool. Random recombination will then have one particular configuration of the heavy chain. Similarly, for the light chain, it happens similarly, but in this case, you have both the kappa and the lambda gene loci. So this is sort of textbook-based immunology. We know that these gene recombination events leads to a huge diversity in the antibody repertoire. And I started as kind of a more fundamental biology sort of question, which is that, okay, I know that each one of this partner, the heavy and light chain, each of them can have millions or thousands of millions of combinations or possibilities within a particular person. The basic question that we have is, if each of them can have such a huge diversity, are there particular rules that tell us that this particular heavy chain and this particular light chain can pair to form a stable antibody, and this particular heavy chain and light chain pairs, the antibody will not work. Now, by not working, there can be multiple reasons. It can be that they are just inherently incompatible with one another. So you picking this heavy chain means that I cannot accept certain combination of V and J genes on the light chain. Otherwise, the resultant antibody will not be stable. But as we all know, during sort of natural development of B cells in our body, you also have other filters that remove, for instance, self-reactive autoantibodies from the system. So I think the sort of biological question we had was, it was quite difficult to actually tease out these two things, right? Being incompatible, or they actually work, but they are binding to the wrong thing. We wanted to see, are there molecular rules that govern, in general, which heavy chain can couple with which light chain? So this is almost like a slot machine kind of analogy that I put, which is that because the heavy and light chain recombine randomly, we want to know, is this simply by luck that this heavy chain will choose this light chain as a partner, or there actually are sort of sequence-based features we can extract from the data to tell us, if you have this heavy chain, you should look at these light chains because they are more stable when form a complex. This is the basic biological question we have, and we had this really for quite a good number of years, way before sort of single cell approaches become popular in the field. But eventually, we get to the point of actually acquiring enough data, enough single-cell antibody repertoire data that allow us to actually consider computational approaches that learn the rules of antibody chain pairing from this data. So this is work spearheaded by a PhD student in the group, Dongjun. What we did is essentially take these barcoded single-cell B-cell receptor data where we have the amino acid sequences of both the heavy and light chain, and because they are both attached with a cell barcode, I can then go back and trace that these two chains actually come from the same cell and therefore very likely to form a stable pair that actually exists as a viable B cells in order to be captured in this data. The problem here is when you think about typical machine learning methods, how are they trained and developed? A lot of the time you need also negative labels. This is the problem for us because we don't really have negative example here. We have not sampled in sort of real life biological systems heavy and light chain pairs that do not work because they would have been removed in B-cell development. So the best approach we managed to get to is to generate these so-called pseudo-negative examples. Essentially, these are just swapped partners. So if in here, in the observed paired chains and data, we have this blue pair and this green pairs, the pseudo-negative example would essentially be swapped partners. And we take this as negative. Now, of course, there is the possibility that these swapped partners may in fact be viable as a pair. This is not something that we can sort of ascertain given what we have. But that to us is the best sort of approximation of what negative will look like in this case. So once we have this positive and pseudo-negative examples, we were then trying to see whether we can obtain classification models, computational models that allow us to distinguish the positive cases from the pseudo-negative cases. So we first tried to see what part of the antibody sequence contains information that allows us to predict chain pairing. The first thing we did was a very sort of naive idea, which is if you just take the V and J gene usage from the heavy and light chains, you build very simple regression or classification trees. How would you be performing in this task of telling apart the cognate pairs from these randomly reshuffled pseudo-negative pairs? So shown on the y-axis are the accuracies of these models. You can see 0.5 is literally flipping a coin, really. You don't do that much better than that. So probably just the gene usage is not enough. I'm pretty sure that this audience would be aware of sort of basics about antibody structure. You have particularly the CDR regions, which are highly variable, and as you can see here, also form quite a bit of contact between the heavy and the light chain. So we thought, what if we don't just use the gene usage, we actually try to do something with the heavy and light chain CDR sequences. So these orange boxes correspond to models built using the CDR sequences. We implemented convolutional neural networks for this purpose. These different configurations are just different combination of parameters you can pick and use in training your CNN networks. As you can see, however you change it, eventually you will reach a plateau of around 0.6 accuracy. So with the sequences, it performs a bit better, but it seems that there's a limit we're hitting. So this is the around the time when this sort of large language models become very, very popular in sort of general natural language processing, but also begin to kind of go into the field of biological sequence analysis. In particular, there are protein language models, which basically borrow the methods developed for embedding natural languages, essentially to have this sort of fill in the blank task to learn the grammar of the languages. There are also protein language models that learn the grammar of the amino acid sequences by going through and learning from a lot of protein sequences. There are antibody-specific versions of these models that has only seen antibodies and are therefore quite good at recovering the antibody sequence information. So we thought maybe if we use one of these antibody language models, we fine-tune these models our data, it may perform better. So this is what we kind of settled on at the end. We call this particular version ImmunoMatch. This is an antibody language model, which I think in this case is antibody-2, fine-tune on our data to predict and classify antibody chain pairing. So this has quite a lot improvement over both the gene usage only models and the CDR only models. And this is the sort of final model that we produced in our work. There are also more validation that we did. For example, does the model still performs well on test sequences that it has never seen during training? We also, in particular, conceived a more difficult test case, which is if I have sequences, antibody heavy and light chain pairs that are sampled from different donors, so from a completely different antibody repertoire, can ImmunoMatch still be able to recover the heavy and light chain pair information? So naturally, this is a harder task, and therefore the performance drop a little bit. As you can see, it's still quite a substantial improvement from the sort of random models that you can see. So in practice, we think that this is quite a good model that we have arrived at in terms of telling apart whether a heavy or a light chain is paired or not. So what do we do with our model? Now that we have something that works, our model provides a score that indicates the propensity, the likelihood that a given heavy and light chain will pair. This score can be used for multiple purposes, and we started from the perspective of really understanding the natural biology of B-cell maturation and development. So we were trying to ask questions like this. Does the propensity for chain pairing change as you move from one stage of B-cell maturation to the next? We know, for example, that you have, in a healthy condition, naive B cells will move in a germinal center, come into contact with the antigen, getting educated to improve the specificity and binding affinity to these antigens, and then ultimately acquire immunological memory. Various part of these processes can go wrong and lead to diseases. So this is the sort of basic biology question that we have, but we also have a more sort of applied antibody design and development question, which is the score useful at all in prioritizing stable heavy and light chain pairs when we are trying to do antibody discovery? Now, no matter if you are the basic biologist or the sort of applied antibody development person, to answer these questions, you need validation data sets. So what we did was essentially to collect a whole bunch of different types of data that we can use to answer these questions. So these includes single-cell B-cell receptors, which are not included during training, particularly those where the libraries were generated from sorted B-cell subsets. So we know for sure that these sequences correspond to naive B cells or memory B cells and all the other different cell types. We also have B-cell receptor constructed from bulk data, and this is where I'm going to spend a little bit of time on in terms of how we applied MyXCR in this particular purpose. Towards the end, I'll also show you an example where we think it's quite exciting to think about antibody discovery from tumor tissues using spatial VDJ sequencing data, where we believe ImmunoMatch also can play a very important role there. So we first looked into publicly available data sets and found single-cell BCR sequencing data from naive B cells, germinal center B cells, and memory B cells. And we use ImmunoMatch to score these sequences. As you can see here, on the x-axis here is a pairing score. So essentially, if the score is above 0.5, the model thinks that this given heavy and light chain pair is a naturally existing pair. You can see here that as the B cells mature, moving from naive to germinal center to memory, the ImmunoMatch pairing score increases. So it seems that this feature about optimizing chain pairing is a natural process that occurs during B-cell maturation. So potentially we can use the model to identify optimized mature antibodies as kind of a screening almost for this type of developability screens to find hits of potential therapeutics. So I also mentioned reconstructing BCR sequences from bulk data, and this is where we use MyXCR for this purpose. We thought that in particular for a lot of leukemia and lymphoma cell lines, we may be able to reconstruct these B-cell receptor sequences from bulk RNA-seq data. So what we did was we had a look at the literature. There's actually this paper from 2018 where they were trying to do a very similar thing. So they took Cancer Cell Line Encyclopedia data, took the bulk RNA-seq sequences out, and essentially applied MyXCR to reconstruct these complete rearranged VD and J recombinations. One interesting thing that they found is that you would normally think that these cell lines are sort of monoclonal sort of architecture within these, but actually in a lot of cell lines, you see quite a lot of subclonal sort of diversity. So each color here corresponds to one particular subclone. You can see they are not all completely blue. You have some of them with quite a lot of subclonal diversity as well, as you can see in these sequences of one of the examples. But anyway, it seems to us that this approach could work. So we were trying to do essentially the same thing, this time focusing on the BCR sequences from the hematopoietic cancer cell lines. As you can see here, once you start to sort of filter for the sort of complete VDJ rearrangement without out-of-frame mutations, without premature stop codons, you are left with quite a few only. So a lot of them are removed in this process, which is also something that they found in that previous paper I talked about. So we also, in parallel, look for published data sets where they report panels of leukemia and lymphoma samples where they have identified the complete heavy and light chains from these cancer cells. So we took these, and in this case, this is a slightly different question from before when we look at the naive germinal center and memory. In here, we're simply asking, in the disease context where the different stages of B-cell developments are perturbed and then subsequently lead to cancers, do I still see this trend of the increasing ImmunoMatch pairing score as you move along this sort of B-cell maturation trajectory? The short answer is kind of yes. You have the less mature stages where we found that the leukemia that is coming from these stages, they tend to have a lower ImmunoMatch pairing score. And as you move along this B-cell maturation trajectory, moving on to the more mature B cells, the lymphoma, corresponding lymphoma stages, you tend to see a high ImmunoMatch pairing score as well. So it seems that regardless of whether looking at the healthy condition or the disease condition, in this case, lymphoma and leukemia, this optimization of the heavy and light chain pairing is a hallmark in the maturation process of B cells. Then it becomes another question of antibody discovery. I was also raising this question earlier. Can we actually use ImmunoMatch to facilitate antibody discovery pipelines? So here, nowadays, single-cell sequencing becomes easier and easier, but we still think there are particular holes in modern technologies used for antibody discovery where we do need ImmunoMatch, a method like ImmunoMatch, to help us prioritize likely heavy and light chain pairs for subsequent cloning and antibody discovery purposes. So one example is spatial transcriptomics. I'm pretty sure many of you in the audience would know about this, where we now have technology that allows us to isolate very small areas within the tissues and basically barcode them with distinct nucleotide barcodes, and therefore you can tell the sequences that come from this particular spot where in the sort of spatial relationship of this tissue they actually map to. The problem is a lot of these technologies, the sort of resolution is not exactly single cell, so you can end up with cases where one particular spatial barcode, the diameter of this little well there is actually larger than the diameter of a typical cell, and therefore you can end with multiple cells at one spot. So essentially kind of a mini bulk question and problem that we have here. So we reason that in this case, ImmunoMatch can help if we're able to isolate VD and J sequences from the B cells infiltrated in the tissues, because the method should then allow us to identify whether this heavy and light chain may actually come from the same cell within this multi-cell little well there. So we took data from a method that is developed to essentially do spatial VDJ sequencing. So this is just an example where they have data from a section of a breast tumor. They were trying to do this sequencing of the heavy and light chains. They have distinct clones, so I'm just kind of mapping them onto the same tissue. So you can see here, for example, chain clone, and this is a light chain kappa clone. Essentially, you want to see if they overlay, do they match with one another? So this is often a good indication that these chains might get paired. So in this original paper where they discuss and describe this spatial VDJ method, they developed a colocalization-based approach to essentially ask, how good is this orange and purple color mapping to one another? So ImmunoMatch does something different because now it has direct access to the entire antibody sequences, is able to identify correctly in this case that this heavy and this light chain do make sense if they form a pair. It gets a little bit interesting when you look at other examples. You can have cases where you can see clearly there is some type of spatial colocalization signal, but their colocalization method was not able to pick it up, so a false negative, whereas ImmunoMatch having access to the sequence were able to pick them up correctly. You can also see cases where, for example, despite the colocalization mathematics work, you can actually see spatially that it doesn't necessarily really overlap one another, the heavy and light chain signal. And ImmunoMatch having access to the sequence information is able to correctly say that these two sequences should not pair. So we think that is an exciting application of ImmunoMatch. I think nowadays more and more spatial technologies are applied to look into infiltrated B cells and try to identify tumor reactive sequences, for example, from these B cells. We think that an approach like this that actually looking at the amino acid sequence can really help in facilitating identification and filtering of potential antibodies that you can clone and subsequently validate as a tumor reactive antibody. So in conclusion, I hope I convinced you that we now have ImmunoMatch as a tractable solution that allows to assess whether an antibody heavy and light chain pairs with one another. I also hope that I have convinced you that this is not just application of a fancy deep learning and AI method onto this problem. It also has practical usage and benefit potentially in antibody discovery. And as I'm trying to sort of impress on you during my talk, I'm interested in both the sort of natural development of B cells and also in an engineered system in terms of antibody design and discovery ex vivo. I think that now we have ImmunoMatch as a method that allows to sort of isolate the problem of chain pairing as one aspect of antibody developability. Potentially, we can then think about looking into different methods that capture different aspects of this developability spectrum that really allows to have a better in silico handle on how do we assess and filter an antibody sequence given these different developability considerations. So in the last minute also, I'll just do a little ad about my lab. As I was saying at the very beginning, I just started my group from this January at UCL Structural Molecular Biology. So my lab is called Cross-Scale Interpretable Omics. So I still do quite a lot of computational immunology work, in particular looking into deep learning AI methods to look into antibody immunity, B cell responses, but also from the perspective of antibody design. And there are also stuff that I do that doesn't sit in the B cell spectrum, but more broadly into thinking about reasoning and interpretation from AI models of omics data. So if this is the sort of thing that interests you, please don't hesitate to kind of drop me an email or a message, and I'm very happy to discuss sort of potential collaborations that we can have. So just want to thank the key people involved in the project. So Dongjun, as I was mentioning, is the main driver of the ImmunoMatch project. I also like to thank Franca, who jointly drove this project with me towards success, and also everyone in the lab and my collaborators that are involved in this journey. Thank you very much.
Great, thank you so much, Joseph.
Thank you.
So really interesting topic, and I'm really glad to see it in fruition. So we'll move on to the discussion portion.
Mhm.
So Stan, I'm sure he has lots of questions for you, and also if you have questions in the audience, feel free to drop it in the Q&A.
Yeah, so I can start with my questions. Yeah.
Yes.
Yeah,
Mm.
well, thank you very much, Joseph. That's super interesting and super nice tool. Um, so I actually have a couple of questions, and we already have had discussion with you regarding some of them. So, but first of all, my first question that I want to ask, so about the data size. So,
Mm.
since you are training on single cell data, we know it's like first of all very expensive. We have a limited amount of public data, yeah,
Yeah.
and in general, challenging to update. Do you think the training data set that you had is enough for the model? Or let's say if you do twice more, if you get twice more data, 10x more data, do you think the results will change significantly?
Uh, okay, so this exact question, I hope you're not one of our reviewers because we had exactly the same question. But anyway, so yeah, so we were asked this essentially, right? So if we have more data, does it change the predictions?
Mhm,
So
mhm,
the final model, I think we used three data sets, and in total, I think it
mhm,
was six donors, which if you think in a sort of
mhm,
generic AI machine learning setting is not a lot. We did an experiment where we tried to find more data sets, but at the end, we pushed to around 10 or 11. The reason
mhm,
why it's so limited was we were quite restrictive in the data sets. We want
mhm,
only B cells sampled from healthy individuals. So we have a lot of things from, for example, people receiving vaccinations or
mhm,
people for autoimmune or cancer and so on. We didn't include them deliberately because we don't really know whether these disease conditions influence whatsoever into the pairing signal at all. So we try to keep it
mhm,
as clean as possible. But
mhm,
anyway, we try to increase the number of people. The results do change. Our concern was we found that you increase to additional data sets. I think in the end we have 5 or 6 data sets. Um, there is a trade-off between the accuracy and false
mhm,
positives.
mhm,
So for us, so the false positive did increase, which is to our surprise, because you would normally think that as the model generalized, it would decrease a little bit. But in our case, at least for the data we included, you include more donors, the false positive problem actually were more serious. So there's definitely a data set specific problem there that
mhm,
needs to be looked more and more. So in parallel, one of the things that we do is to try to collate and collect and build this large atlas, both on the transcriptomic side and
mhm,
also the BCR side. So hopefully, I'd like to see a subsequent sort of refresh of ImmunoMatch if we train with more data. Does it still behave like this, or
mhm,
is there a more intelligent way to select which donor should I include in my training? So that would be
mhm,
something that I will be quite interested to look into. But yeah, it does
mhm,
affect it, basically.
mhm. And immediate follow-up. So since you
Hmm.
mentioned uh, you deliberately focus on only on the healthy donors.
Hmm.
Why? Do you think that let's say when you mention a natural pairing, yeah,
Hmm.
it means that if there is a patient with a condition or with a disease state or after vaccination, those B cells are still natural. They're produced by the immune system, no?
If it's vaccination, yes.
Hmm.
But I would argue it probably
Hmm.
depends quite a lot on the sort of background of the donors.
Hmm.
I was particularly um, wary of the autoimmune patients.
Hmm.
Yeah, so it's a natural B cell response, but in particular for those cases, you would think that somewhere in the natural B cell development, there's something wrong in their selection, in their negative
Hmm.
selection of the sequences, and therefore we really don't want to include them. Cancer, I
Hmm.
think, is a similar story, and therefore we
Hmm.
didn't really want to include them. I think there's an
Hmm.
argument potentially about the vaccination side of things, whether we can relax a little bit and we still
Hmm.
see a good signal there. This is something we haven't tried, but I suppose it's one of the things that we want to have a look if later on we decide to sort of update ImmunoMatch with more data.
Thank you very much for the deep answer, and transitioning to the next topic of my
Mm,
question. So, and also related to the data size. Let's say how now on the engineering side, and we work in our company a lot with industry people from biotech and biopharma who do antibody discovery. Yeah, and they're looking for antibodies, especially in the in vivo studies. They're looking for more mature B cells to select them and then to further downstream, etc., etc. So let's first of all a technical question:
mm.
how sensitive ImmunoMatch to, let's say, one amino acid mutation? So if you change one amino acid, will the score change like dramatically, or it's
It
like normal?
can. It can.
Mhm,
Depends on
mhm,
where the mutation is. We did an experiment. I think it's also in the paper where we
mhm,
were trying to see exactly this. So if you have differences, amino acid differences to, let's say, an approved therapeutic antibody, how different would the immunomatch score be? The answer is if, let's say, your amino acid difference actually hits at the heavy and light chain interface, it can matter a lot. Uh,
mhm,
we found actually that a lot of those cases where the differences are huge in the immunomatch score, a
mhm,
lot of those amino acid differences actually map to the interface. But of
mhm.
course, if you want to actually use it as, I can imagine, sort of an in silico mutational scanning type of
Yeah.
idea, uh, a lot of the positions, because they are far away from immunomatch, you may see some differences, but I'm unsure as to how much you can trust them. It really depends on
Mhm,
where the amino acid difference is.
Yeah. Well, got it. Thank you. The further question exploring this like antibody discovery side. Yeah.
Mm.
Can ImmunoMatch be used kind of a score to select better antibodies from, let's say, in vivo discovery campaign, where you sort the
Mm.
cells and then looking something you know, which is interesting.
That's the goal, right? That's, I think, one of the
Hmm
original idea we want immunomesh to be,
hmm
right? So you can actually incorporate them in these
hmm
sort of
hmm
pipelines.
hmm
Um,
And mhm,
so I think you can. There is still, as you can see from this naive versus memory comparison, the
mhm,
score is related also to that. So I
mhm,
think in reality, a lot of these different aspects of developability are kind of interlinked. And
mhm,
but you can potentially definitely use this, but it's not something that we have sort of thoroughly tested how good it is
mhm,
in kind of a real life
mhm,
discovery
mhm,
campaign, I suppose.
mhm.
Yeah.
Yeah, well, from my perspective, since I work a lot with folk who are doing this antibody discovery, it may be like a really interesting thing because the question you have like 10,000 of cells, you need to select 100 or 200 for the downstream functional validation. How? Yeah, what are the best criteria to select them? It's all like you can filter out the liabilities, that the easy ones
Mm,
like sequence liabilities or even
yes.
functional liabilities, but still you have plenty of them. So how to prioritize? And this may be a really good hint or a really good ranking
Yes.
score to prioritize them. So
Yes. Yes.
yeah, and related question again to this, I'm constantly getting this question: is there a way, and that's why I'm thinking about the immunomatch in this sense, is there a way to just do bulk sequencing and then use some nice AI tool to pair them without doing single cell, which is expensive?
Again, was one of the original idea of
Mhm,
the
mhm,
purpose of the tool.
mhm,
We had the idea of ImmunoMatch way before labs
mhm,
start to use single cell to look
mhm,
at antibody repertoires. So that's the idea. In theory, you can use ImmunoMatch to pair sequences. It's not something that we have sort of
mhm,
thoroughly tested when we
mhm,
did the work and published the paper. Um,
mhm,
I very much hope that we can have a look at that and see whether that's the case. Because
mhm,
if that works, you
mhm,
benefit from the depth of sampling in the bulk, which
mhm,
is a very
mhm,
powerful thing when you want to discover things, as
mhm,
opposed to
mhm,
being limited by the number of cells you can capture.
mhm,
So I think there is great potential there. It's something
mhm,
that needs to be
mhm,
validated.
mhm. Yeah, I actually recommended ImmunoMatch to one scientist who was looking to reconstruct the paired heavy light repeater from bulk RNA-seq data when you don't have a lot of clonotypes. So you just
Yes,
you can combinatorially like easily combine them and see like where the best score, and then most probably this is your like real
exactly. Yes.
antibody. Yeah, so it's a good use case. And Monica, I think we have questions from the audience.
Exactly. So,
Yeah.
Joseph, we have one question. Did you try using a general protein model like ESM2 instead of anti-beta-2? I found that it generally works better than antibody-specific models, especially with relation to structural features.
Yes. Short answer is yes, and it's in the paper. We basically tried ESM2, which I think is generic 650 million parameters ESM2 with anti-beta. At the anti-beta version, fine-tuned performs better than ESM2, and therefore that was the final version. But actually, we found that in parallel work, I agree with that person who asked the question. When you are interested in the structure features, it seems that ESM2 is a bit better at capturing those things. Number two is the ESM2 is also more receptive towards fine-tuning in the sense that the difference between the raw ESM2 versus the fine-tuned is a lot bigger in terms of performance jump compared to before and after fine-tune of anti-beta 2. So it's something that we found. There is something, I think this idea of the utility of antibody language models in this type of things is a very interesting topic. And I'm not sure that we fully grasp the idea of when do we use what and how do we use it. So that's something that I think is a very fascinating question. But short answer, we did try. But
Okay, and next question. Nice presentation, Dr. Ng, as you alluded to as well about the CDR context of FRW. Our other determinant of pairing recently published by Natural Antibody publication from Comrade. Does your model capture some of the canonical residue and interaction?
Yes, so we try to do things like I don't know this person who asked the question know about sort of mechanics of the language models. One of the things you can try to extract from the model is so-called attention between the chains. So that means I put the heavy and light chain in, and you can actually extract from the model which pair of positions it actually look into towards making the predictions. And you can then try to see are there contact that resemble, let's say, CDR contact or CDR framework contact and whatnot. We try to do this. It becomes quite difficult once you grasp the idea that there are multiple branches in the same model that would generate something like this. So we find it quite difficult to capture, but what I can definitely say is we talked about this idea of mutational scanning a little bit, right? You can have cases where ImmunoMatch will give you a very big score jump if you have mutations that are not really in the CDR, but it's also in the contact between the heavy and light chain within the framework. So we think that there are definitely signals that is not CDR specific, that after this fine-tuning exercise that we have to make ImmunoMatch was able to capture. We also tried in parallel to see, forget about this AI and language model stuff, but we just start from existing NLP structures. Are you able to extract anything? It was quite difficult, as you can imagine, simply because you are limited by quite a lot in terms of the number of NLP structures you can get to look at these type of contacts. So short answer is yes. There are things there in the framework that tells you about whether the pairing is viable.
And then next question, if I wanted to get access to ImmunoMatch, where can I find it?
So it's on the GitHub. So I think if you Google ImmunoMatch, you should find either the GitHub or the paper. So we have on the GitHub code that allow to use the model. So the fine-tuned model version is on Hugging Face. On the GitHub, we also provide a code notebook where you can use you can upload data and then it can score for you. But yeah, so the code is publicly accessible.
Great. Next question. Thank you for the great talk. Did you look at the correlation between SMH mutation rate with ImmunoMatch score?
Yes. So in this naive versus memory comparison, we also have the somatic hypermutation rates. As you would imagine, because there's a difference between naive and memory, the mutational level also correlate with the ImmunoMatch pairing score.
Okay, great. Stan, you have any other questions?
Yeah, I have actually a few other questions.
Yes.
So one is particularly interesting for me. Is B-specifics. So do you envision to let's say use the learnings that you or the model that you built for ImmunoMatch for the opposite, not to predict the natural pairing, but to design the orthogonal, antibodies?
Would be very cool. We didn't think about that, but would be good. I did remember this type of conversation about biospecifics and so on when we talked to, the more antibody discovery side of, people working on that side of the coin, really. I think it will be very cool. I think the strength here is because ImmunoMatch is trained on normal antibodies. I think that is quite powerful in terms of actually thinking about in these sort of more sophisticated engineering sort of forms of antibodies. I think there's a use of it. Yeah, definitely.
So, and we also have one more question in the chat, and it's related to my next question. So the question from the Li He is, do you think that ImmunoMatch is biased towards the human species? And my follow-up question immediately, it's related to the question I asked about one amino acid mutation, is can you use ImmunoMatch for humanization purposes? And let's say when you're optimizing the antibody toward the human scaffold, you're looking on those like mutations, difference that you need to optimize to see like how it will drop in the score, because the developability and the affinity may really drop to the parts.
There is, I think, the bias bit that's I don't know. Anecdotally, when we try mouse data on ImmunoMatch, it is able to capture what is a pair and what is not. So you can probably say there is some form of generalizability that ImmunoMatch has already learned. Only look at the human data about whether heavy and light chain pairs. But the truth is, I don't think we systematically try this and see what is the level of trust you can put on ImmunoMatch on the mouse data. The other bit about using it for humanization, I think also links to this. Until we have a handle about the uncertainty around human data and mouse data on ImmunoMatch, I'm not sure how much you can trust the ImmunoMatch suggested mutation, say, as a humanized version. I still think it's a gray area because on the one hand, all of these objects, whether human or mouse, the pairing, I mean, if you look at the structure, they assemble in a very similar way. But on the other hand, there are substantial sequence differences between them. So I don't know the exact answer to that. Is the honest answer.
And by the way, the other question that we are getting from Le here is, are you, Monica, you already closed the question. I'm trying to read. So
sorry.
are you keep on adding more training data to improve the model?
I think this is something we wanted to do. It's also the sort of thing that as an academic like me, we're starting, it's a sort of thing that you can give a student to do, to look for data and then try to fine-tune the model. I think it's important, and I would be interested to see, is there a principled way that allows us, as you were alluding to in our first discussion points, right? How do you tell adding the data, adding additional training data help the model or not? So I think if we have a principled way to say, adding this is helpful, adding this other data set is not helpful, that would be great for ImmunoMatch on its own, but also in a generic sense about applying AI methods to predict antibody properties, right? Because then you can have a rational way of building your data set to train a model rather than just, grabbing everything you can from public spaces.
Yeah, I agree with you actually.
Yeah.
So, Monica, if we have time for one more question, Monica,
Yeah,
can I ask one more question?
yeah,
Uh,
absolutely.
so, returning to the developability,
Yeah.
I just curious. I tried to play this myself slightly, but didn't have enough data to check. But obviously, you're focusing on the natural pairing, so it's not about our problems with the manufacturing of antibodies. Yeah, those are different problems. But did you try to look at the let's say liabilities and correlation between the well-known liabilities that we know will fail downstream manufacturing with the ImmunoMatch score?
We did not. We actually have another student who were trying to do similar things, but we never tried to put ImmunoMatch into the equation and ask how these things correlate. I'll be quite interested to see. There are also measurements about like just in general specifically. So I think we tend to conflate these things, right? When we are measuring, let's say, the thermostability of the antibody, we typically think, okay, of course, the pairing is one aspect. But if, let's say, you have a amino acid substitution somewhere in the middle of the immunoglobulin of the heavy chain, it doesn't matter the pairing at all. I think in practice, it's kind of a multifaceted problem, a lot of these developability things. So it would be quite interesting to see how does ImmunoMatch combined with all these other metrics actually tell you about whether something is developable or not.
Super. Well, thank you very much, Joseph. Monica, I can continue asking like lots of questions. So it's so interesting.
Thank you very much. Thank you.
Okay, great. Thank you so much, Joseph, for your time. And thank you for everyone in the audience. We'll be sending out this recording afterward. And thank you so much for your participation tonight.
Thank you, Joseph.
Thank you. Thank you very much for inviting me.
See ya.
See you. Have a good evening.